Right. Okay. Okay. My name's Alexei Drummond. I'm from the University of Auckland in New Zealand, and I just want to introduce a very new genomic observatory called the Hauturu Genomic Observatory. This is a little offshore island in New Zealand. And just by way of introduction, it's a small island, 30 square kilometers, about 1.5 million years old, volcanic in origin. And it's about 80 kilometers northeast of Auckland, which is the main city in New Zealand. It's regarded as it's a restricted site, so there's only a ranger living on there permanently and scientists that work in facilities that can house maybe up to 15 or 20 people. Otherwise, it's uninhabited since about 100 years ago or so, and it's a very important conservation site for New Zealand, as it's sort of one of the last remaining reasonably untouched and modified areas of terrestrial forest, and it's a source for many of the translocation efforts we make and conservation efforts. So it's a key site for us, and as a result, we really want to understand it. There's been long-term ecological research done on this site for 50 or 60 years in community ecology, as well as long-term studies of invertebrate diversity and also phenology and things like that. But the genomic aspects of this of research on the island have only been begun very recently. So the main objectives of the project that we have that we're running through the Alan Wilson Center, which is the main institutional partner. It's a center of research excellence that spans most of the universities in New Zealand and a number of the Crown Research Institutes, is to develop Little Barrier as a model ecosystem. Similar to what they're trying to do on Moorea, but we came at it from the perspective of how can we study an ecosystem without having an inventory of everything, and how can we do this in a cost-effective, affordable way that we could reproduce in other places more broadly. So as a sentinel site, but developing techniques on it that we could spread out to understanding the rest of New Zealand initially, especially terrestrial forests. So next-gen sequencing sampling is the approach we want to take, and as much as possible getting information about the entire ecosystem just out of soil and leaf litter and those kind of substrates via next-gen sequencing. So, I mean, obviously this involves developing some statistical tools as well to understand that data and to integrate information across different communities, so from microbes through to the mesofauna, the invertebrates, up to the birds and the trees. So we're interested in a predictive model as well, that what we develop on Little Barrier, we will then be able to apply to understand what we should be doing from a conservation and restoration perspective in other parts of New Zealand. Up until 800 years ago, the entire North Island of New Zealand had very similar forests to what are out on Little Barrier. They've been mostly removed and replaced by agricultural and urban land now on the mainland, but there's still some regions left that we want to try and conserve and restore. How best to do that? So this is Little Barrier in the context of the North Island of New Zealand, circled. And it's similar in environment type to a number of other key regions on the mainland that are conservation efforts. So this is why we think it's a good model. These are it's a plot-based sampling procedure, so we're sampling about 30 or 40 plots intensively within the island for the initial pilot project. And in each of these plots, we identify all of the trees, the bryophytes and the lichen species, and we do a next-gen sampling of the soil for a number of taxa. And this is just an example of some of the phylogenetic data that we obtain from these plots. We've also initially built a reference data set of where we've collected invertebrates and done some morphological identification, and we have a reference data set, but we want to eventually go to a completely DNA approach, so that's really just for validation purposes. And there's a number of people involved in the project, so it's really a cross-institutional effort that includes key stakeholders like the Auckland Council and the Department of Conservation. So thank you very much.